CRISPR revolutionized genetics by making precise DNA edits faster and cheaper. In this video, we'll explain the molecular basis of CRISPR, how researchers use it across fields, real examples, and the technical and ethical challenges ahead. Chapter 1: Origins and Basic Biology. CRISPR originates from clustered, regularly interspaced short palindromic repeats discovered in bacterial genomes in the 1980s and later recognized as an adaptive immune system. In 2012, Jennifer Doudna and Emmanuelle Charpentier adapted the CRISPR-Cas9 system for programmable genome editing. The bacterial mechanism uses short RNA guides of about 20 nucleotides complementary to target DNA, and a nuclease, typically Cas9, that requires a protospacer-adjacent motif, such as NGG. When the guide RNA pairs with matching DNA, Cas9 creates a double-strand break. Cells repair these breaks by non-homologous end joining or homology-directed repair, allowing gene disruption or precise sequence insertion. Chapter 2: How CRISPR actually works. At molecular level, CRISPR editing relies on two components: a programmable single-guide RNA of roughly 20 nucleotides that matches the genomic target, and a Cas nuclease that binds the guide and cuts DNA. Cas9 contains HNH and RUV-C nuclease domains that together cleave both DNA strands, 3 base pairs upstream of the PAM. Successful binding requires the PAM sequence, constraining target choices. After cleavage, cells repair the break. Non-homologous end joining often introduces insertions or deletions of a few base pairs, while homology-directed repair can incorporate donor DNA, but is less efficient, typically under 10% in many cell types. Newer tools like base editors or prime editors avoid double-strand breaks to reduce unintended edits. Chapter 3: Real-world applications and examples. CRISPR has moved from lab to concrete examples across medicine, agriculture, and diagnostics. In 2016, researchers produced a non-browning mushroom by knocking out a polyphenol oxidase gene. US regulators did not impose GMO rules. In diagnostics, CRISPR-based platforms such as Sherlock and Detector were adapted in 2020 to detect SARS-CoV-2, delivering results in under 1 hour. In therapeutics, ex vivo editing of hematopoietic stem cells, notably CTX001, developed by CRISPR Therapeutics and Vertex, entered clinical trials for sickle cell disease and beta thalassemia, reporting patients free of transfusions or crises in early-phase studies. Agricultural yield, disease resistance, and model organism research are expanding rapidly. Chapter 4: Advanced Challenges and Ethical Implications. Advanced challenges determine CRISPR's future. Delivery remains central. In vivo approaches use viral vectors, such as adeno-associated virus, with a packaging limit around 4.7 kilobases, while SP-Cas9 coding sequence is roughly 4.2 kilobases, prompting smaller nucleases or split systems. Lipid nanoparticles enable transient delivery and are used clinically. Off-target edits and immune responses to bacterial nucleases require sensitive detection methods like guide-seq and targeted deep sequencing. Newer modalities, base editors and prime editors, developed since 2016 to 2019, perform single base conversions or small insertions without double-strand breaks, reducing collateral damage. Ethical limits are critical after the 2018 germline editing of human embryos, which drew universal condemnation and tightened regulation worldwide. CRISPR transformed biology by converting a bacterial immune system into a precise genetic toolkit. Viewers should remember core components: guide RNA, Cas nuclease, PAM, repair pathways NHEJ versus HDR, major applications in medicine and agriculture, and the technical and ethical challenges shaping responsible deployment.